All right, good afternoon. We are ready to start. My name is Yannis Paskalidis. I'm one of the faculty here. And this is part of our distinguished seminar series at the Hariri Institute. And it's jointly organized with the Machine Learning in Medicine seminar series. This is a seminar series that we are co-organizing with the University of Pittsburgh and the University of Toronto. And some of the talks are in person at these institutions, and there is also a crowd that is online. So the talk today is hybrid. So instead of introducing the speaker, we'll sort of do a Markov chain transition. I'll introduce Archana Venkataraman, who is one of our new faculty in electrical and computer engineering here at BU. We're able to steal her from Hopkins to join us. Just a few notes for those of you that are here. Please hold your questions for the end, unless you have a clarifying question that it's really burning. I guess you can interrupt, but hold the more significant questions for the end of the talk. And for those of you that are online, feel free to use the Q&A function of Zoom, and we'll be monitoring and we'll bring the questions forward at the very end. So with that, Archana.
Thank you, Yannis, for the introduction. So today it's my pleasure to introduce our speaker, Professor Dani Bassett. So Dani Bassett is the J. Peter Skirkanich Professor in the departments of Bioengineering, Electrical and Systems Engineering, Physics and Astronomy, Neurology and Psychiatry at the University of Pennsylvania. They are also an external professor of the Santa Fe Institute. Professor Bassett's work blends neural and systems engineering to excavate mechanisms of cognition and disease in human brain networks. Professor Bassett has earned numerous awards and recognitions, including an Alfred P. Sloan Research Fellowship, a MacArthur Fellow Genius Grant, an Early Academic Achievement Award from the IEEE Engineering in Medicine and Biology, or EMBS Society, an ONR Young Investigator Award, an NSF Career Award, being named one of Popular Science's Brilliant 10, a Lagrange Prize in Complex System Science, and being inducted into the AIMBE College of Fellows. So we are honored to have Professor Bassett here as our speaker and excited to learn more about understanding brain network modulation.
Wonderful. Thank you so much for the opportunity to come be with you all here today. I'm excited to tell you about understanding brain network modulation over the next few minutes. Where I would like to start is by observing that many biological systems are organized as networks, and the functions of those biological systems are made possible by the pattern of connections among units, where items of interest like nutrients or goods or information can be routed. If we look at this example, we can see that organs are connected with one another. Inside of organs, there are cellular networks. Inside of cells, there are molecular networks, and genes beneath that. But even if we go beyond the subhuman or intrahuman networks, we can observe networks between individuals in the context of society. So on the right-hand side, you see an example of a social network. In each of these cases, there are different sorts of connections between items that allow for different sorts of things to move, as I said, nutrients or goods or information. I wanted to start there because I want to think about the brain as a particularly good example of a networked biological system where that network architecture really matters for how the system functions. So the human brain is comprised of regions that perform specific functions and engage in particular computations. Here, for example, what I'm showing you is a pattern of activation on the surface of the cortex when somebody is moving their finger. So this highlights an individual set of regions that support motoric function. On the left-hand side, what I'm showing you is a set of brain regions that are defined by differences in activation, differences in cytoarchitecture, and differences in structural connectivity. Each of these regions can support different sorts of functions like motor function, vision, audition, object recognition, attention, decision-making, emotion processing, language, and cognitive control, to name a few. In humans, these functions are often studied using non-invasive imaging techniques like magnetic resonance imaging, and in particular, functional magnetic resonance imaging. So that helps us to think a little bit about what the pieces are that comprise the brain. But now I want to think about what the connections are. How are these pieces connected to one another? And how does the pattern of connectivity help us to understand how the system functions? Brain regions are connected by large-scale white matter tracts, and each tract is a bundle of neuronal axons along which information-bearing electrical signals can propagate. Here, what I'm showing you is an image from a diffusion magnetic resonance imaging scan, and then I'm blowing up that picture or zooming in to show you a pattern of connections between different units, and then I'm zooming in even further. So these signals, these electrical signals, can be intrinsic, so related to neural processing, or they can be extrinsic, driven by external applied stimulation. So if we think about this structure that we can then see in individual humans that changes or varies across different humans, we can begin to understand that the structural constraints imposed by this pattern of connections is necessary for us to unpack in order to understand both healthy brain function, but also the response of the brain to external stimulation. That becomes really relevant when we're thinking about medical interventions, such as brain stimulation for the control of epileptic seizures or for the treatment of other conditions. So what I want to focus on in the next few minutes is thinking through these structural constraints in the brain. Why has the field sort of moved to try to understand these structural constraints at a deeper level recently? How has the field done that? And how does that help us to understand the impacts of stimulation in humans? So the recent efforts in this area have expanded the investigation of structural constraints in a couple different ways. The first one is that we now have increasingly sensitive measurements. So where a decade ago, we were able to scan using diffusion magnetic resonance imaging, we were able to scan the brain and understand the pattern of connectivity at a relatively coarse scale. Now, with increasing scan times, but also an increasing number of diffusion directions and a variety of expansions on the algorithmic side, we're able to see the pattern of connectivity at a finer and finer resolution. So we are now increasingly sensitive to microstructural integrity, and we are able to locate the tracks that exist in the brain at finer and finer spatial resolutions. So we end up getting a picture that's like this, for example, which every time I see it, I can't decide whether it's a shag rug that I really want to like step on, or a puppy wants to play on, or the brain. This one is the brain, but you could imagine creating such a rug. Okay. What is the second way in which our understanding of structural constraints has changed recently? Well, over the last couple of years, there's been an increase in data-informed computational models that take into account this pattern of structural connectivity and seek to understand how that pattern relates to the system's functions. So we are now able to quantitatively assess how any particular network architecture, so the one in my brain versus the one in your brain versus the one in our friend's brain, affects the brain's potential or actual dynamical repertoire and its response to stimulation. So increasingly sensitive measurements and also data-informed computational models. But there's a third key advance over the last few years, which is that there's been a change in our conceptual paradigms that we're bringing to these data. In earlier work, there was a focus on brain mapping, on understanding which regions are being activated and how much, and why. And increasingly, we are also trying to use structurally based explanations to understand patterns of activity and repertoires of dynamics. So this is not to suggest that activity-based explanations are inaccurate or wrong in any way, but to suggest that to understand human brain dynamics, it is increasingly useful to combine activity-based explanations and structure-based explanations. And that combination allows us to think through how these regions are activated in relation to one another. So these three changes over the last few years, these changes in measurement, in modeling, and in conceptual explanations are providing a richer understanding of structural constraints on neural function and on stimulation. Now, you might be thinking, okay, this is all great, but it's pretty high level. Let's work through an example. So I want to first focus on an example in the third area, a conceptual shift in explanations for intervention. And then after that, I'm going to move to the second area, data-informed computational models, and then I'll close. So first, thinking through a particular example in this conceptual shift in explanations from activity-based explanations to structure-based explanations. This example that I want to discuss is one that focuses on the use of transcranial magnetic stimulation, or TMS. The work was led by graduate student Val Sidner and in collaboration with Ted Satterthwaite, who is her immediate supervisor, and also Desmond Othis, who is well known for his work combining TMS and fMRI. It was recently published in Science Advances. So in this study, 45 healthy participants underwent a TMS protocol where TMS was applied to the ventrolateral prefrontal cortex, or vIPFC for short, which is known for its role in emotion regulation. That TMS was interleaved with functional MRI. So you can see the structure of the experimental setup up here. So we have single pulse TMS fMRI interleaved. After that, we did event modeling and then quantified the evoked response, so the signal change in the bold response in a particular area. Separate from the TMS fMRI data acquisition and application, we also collected diffusion MRI data from these individuals and applied a pre-processing stream that is particularly robust to motion artifacts and was recently developed. From that, we extracted a whole brain tractogram, or the pattern of inferred structural connections between brain regions. And then we also estimated the fiber density between two regions of interest. So these are the two types of data and two preprocessing streams that allowed us to acquire information about the change in the bold signal after a single pulse of TMS and the fiber density between particular regions, or the structural constraint on where that signal might be transmitted. What we find is that stimulation to the ventrolateral prefrontal cortex evokes amygdala activity. And this is done in an individually specific way. So on the left-hand side, what you are seeing is individual areas or sites of stimulation for individual people. And then here, what you're seeing is the evoked bold response. So this is in the left amygdala. There is a change in the bold response after the TMS to the ventrolateral prefrontal cortex. So I want to make sure that this is clear that these are two different regions. We are stimulating vIPFC, and then we're measuring changes in the bold signal in amygdala. And those two regions are structurally connected to one another. What is the point of view? So it's just highlighting, that one's highlighting the amygdala. Yeah. And then, so interestingly, there's a strong variation in the degree of bold signal change across participants. So each of these data points is a different person inside of the experiment. And what you can see is that the 0 line sits here. So what that means is that when stimulation is applied to the ventrolateral prefrontal cortex, amygdala activation can increase or it can decrease, and that depends on the person. Now, we also want to know whether the amygdala, the change in the signal in the amygdala is larger than expected or less than expected in other subcortical areas. So that's what you're seeing on the right-hand side of this figure. So this is the difference in the TMS evoked response magnitude At the 0 line is where the amygdala activation would be. So anything above the 0 line means that the evoked response magnitude in that region is larger than it was in the amygdala. And anything below the 0 line indicates that the evoked response magnitude was smaller in that region than it was in the amygdala. On the x-axis here, you have abbreviations for the different subcortical regions, including caudate, putamen, hippocampus, thalamus, etc. So here what you are observing is that the majority of these regions have activations that are, the magnitude was larger in the amygdala than in these regions. And there's only one region where the evoked magnitude was smaller in the amygdala than in that region. So what that suggests is that the TMS signal elicited a sizable decrease in the fMRI bold response in the ipsilateral amygdala, and that evoked response magnitude was greater in the amygdala than in most of the other subcortical structures, indicating for us some spatial specificity of where this stimulation is impacting the system. So now the question is, can we understand some of this variation in human responses to the stimulation based on individual variation in the structural connectivity between the region that's being stimulated and the region whose signal change we care about, which is the amygdala? So to answer that question, we calculated the fiber density in this pathway between the ventrolateral prefrontal cortex and the amygdala. So here is that pathway. So you can see the individual diffusion directions and then the extraction of the fibers. From that, we can calculate the fiber density, and we can correct it for age, because age is an important mediating variable in the degree of fiber density between these two areas. So here what you are seeing is the left amygdala TMS evoked response magnitude, and along the x-axis is the fiber density. Every data point here is a different person in the experiment. So here what you see is a positive connection between these two variables. What does that mean? That means that for the participants who had the largest changes in the amygdala signal, those are also the ones that have the greatest fiber density between the region that's being stimulated and the amygdala, where we're calculating the signal change. So that means that fiber density, or the structural constraint, the structural pathway between these two regions, is an explanation for how much that region inside of the brain is going to be activated. Now, on the right-hand side here, you're seeing a separation of the data into two groups of individuals. I had told you, or pointed out on this slide, that some participants have a positive change in the amygdala signal, and some participants had a negative change in the amygdala signal. So what we're doing is replotting the data with those two groups separated. So here along in the lighter pink is one set of individuals, and in the darker pink is another set of individuals. So by separating the data in this way, we can see that the higher fiber density was associated with a bold decrease in the negative evoked response individuals. So that's this dark purple, and a bold increase in the positive evoked response individuals. And so that's the lighter purple. So there is significant individual variation. There's the variation across these two groups is also highly dependent on their fiber density between these two regions. So a simple summary of this example, which is obviously a much broader study, but I just gave you some of the highlights, is that the work provides evidence of amygdala engagement by TMS, so by transcranial magnetic stimulation, highlighting stimulation of this vIPFC amygdala circuit as a candidate treatment for transdiagnostic psychopathology. But more broadly, it also indicates that targeting cortical-subcortical structural connections may enhance the impact of TMS on subcortical activity, and by extension, subcortical-subserved behaviors. So with that example in mind, I now want to turn from this structure-based explanation for how stimulation impacts the system to this second point, which is the use of data-informed computational models. So now understanding that stimulation can flow along these structural connections in a way that's heavily individual specific. I want to broaden out from a single pathway to all pathways, all structural connections in the brain, and think through what kind of computational models we can use to understand the flow of signals in that system. One kind of computational model in this space that I'm particularly excited about is the network control model. The network control model is one that has been developed and applied extensively in systems engineering and has been applied in the context of robotics, the power grid, communication systems, air traffic control systems, and lots of other examples. But the basic idea is that we have a network. In this particular example, this is a very simple network of 4 nodes and 4 edges. The arrow indicates the direction of that edge. And that system can have exogenous input placed into it. So these purple squiggly lines indicate exogenous input. That exogenous input is defined as U, or our notation is U, and can vary across time. So this is a time varying input injected into a particular region or node in the network that can impact the state of all of these pieces. So how exactly does it impact the state? Well, here's an example of a 4-dimensional state space where the initial state was at the origin. And then after the input of these 2 signals, the final state is somewhere over here. One of the questions that is frequently asked in the context of network control models is, what stimulation should I inject, or what input should I inject? And what kind of trajectory would I desire? What should be happening in the middle here to move the system from an initial state to a desired final state? So this is a generic problem and I've been trying to describe it in a very generic way on this slide. But what I'm really interested in is how we can apply that kind of model and ask those sorts of questions in the context of the human brain to better understand the impact of stimulation. So the network control model depends upon 2 factors. One is a understanding of which pieces of the system are connected to which. So this adjacency matrix A is a pattern of connectivity, or the structural constraints in the system. Here, in the context of the brain, we're going to use tractography approaches, as I've been describing in the first section of the talk. But in addition to understanding which pieces of the brain are connected to which and by how much, we also need to understand how activity flows through the system, or have a hypothesis about how the dynamics of the activity progress. So we need to write down a model of dynamics. One very, very simple model is this one, but we can expand this in interesting ways, and I'll touch on that later. So this very simple model of the dynamics suggests or states that the state of the brain, X, at time t+1 is equal to the state of the brain at time t, so the moment before, times the adjacency matrix, or the structural connectivity between brain regions, and then plus the control. So B sub kappa is a matrix, typically with 1s along the diagonal, indicating which regions are actually having the control input injected into them. And then U sub kappa is the control input that changes as a function of time. Note that this is a linear model. It is also time invariant in the sense that the adjacency matrix is not changing. So the pattern of structural connections is not changing. If we were to consider a spatial resolution at which structural connections change a lot, so at the level of synapses, for example, we would want to have A be a function of time. There is also currently no noise in this version of the model. And so if noise is really important for what we're trying to explain, then we would want to add that. So you can think of expansions to this simple model. But to explain how this model works in pictures, what we have is individual brain states, which are patterns of neural activity. So these are vectors from of size 1 by n, where each element indicates how much activity is present in that region. If we are applying this in the context of non-invasive imaging in humans, that activity may be from the bold signal from functional magnetic resonance imaging. If we're using electrocorticography, that might be a power in a particular frequency band. And the idea is that the brain moves from one pattern of activation to another pattern of activation in a way that's heavily constrained by the structural connectivity between those areas. And importantly, what we're really interested in asking is how much energy, how much input is required to move the brain from this state to this state, given which regions are connected versus which regions are not connected. So that energy is going to depend on the regional connectivity. Okay, so let's walk through this in a couple examples, and then I will add some expansions of the model and some caveats. So to walk through this idea a little bit further, what I'm interested in is understanding the relationship between the pattern of structural connections between brain regions and the pattern of activity. So I'm going to be thinking in couplets now. I'm going to be thinking in couplets of activation states and the structural network underneath. So we know that active neurons or neural units can spread activity to other neurons or neural units around them, and that therefore having a whole active population is easy if the neurons in that population are connected. That helps us to think through which couplets are easy versus difficult. So here are a couple examples just in pictures. Here, for example, if you focus on the top left, is a structural network that is modular. So the first 5 units are structurally densely connected with one another. The second 5 units are densely connected, and there's only one line in between these 2 modules. The state that I want this system to go to has activation of the first 5 units and deactivation of the second 5 units. Now, I'm saying that this is an easy couplet because I can send a sort of generic excitatory signal into the first module and a generic inhibitory signal into the second module. And because there's only one line in between, that excitation and inhibition can stay separated pretty easily. So that's why it's easy. And I want to contrast that with these 2 other examples where this state is very difficult for this network to attain or to maintain for any period of time. So here you see a network where we have the modules again, but now they're densely interconnected with one another instead of being sparsely connected. And what I'd like that network to do is to hold activation of the first 5 units and deactivation of the second 5 units. That's really hard because as soon as I activate the first 5, that activity can flow along these structural connections over into the second module and activate it. So I would need to control all of these connections or continuously inhibit this one and continuously excite this one in order to allow this system to reach that state. So it requires more control over a more extended period of time and possibly more complex control by controlling the edges or the connections rather than just the regions. Here's another example of a difficult couplet where we have the modular network from the top, but the state I want it to go to has activation of the first unit, deactivation of the second, activation of the next, deactivation of the next, etc. So this binary kind of on-off state. This is really hard on this network because I can't take advantage of the structure at all. Here I could take advantage of the structure to get to this state. Here I have to fight against the structure. Same here, I have to fight against the structure. Fighting against the structure means injecting more control inputs, more complex control inputs, more spatially distributed control inputs, etc. Okay, so that understanding of how a network can support a particular activation state also helps us to think through just intuitively how a system could move between 2 states. So here, for example, I'm going to talk not just about one state, but about 2, and I'm going to ask how might a system move from this state to this one if it sits on this network. So here's the modular network. I told you it was pretty easy for it to attain and maintain this state. But now what I want to do is to change such that anything that was on initially is now off and anything that was off initially is now on. To do that, I can inject an inhibitory control signal into the first module, an excitatory control signal into the second module, and that will allow me to make this switch. So 2 control signals. In contrast, a really difficult state transition would be the one that I'm showing you here at the bottom. Ignore the fact that it's even hard for this matrix or this network to get to the first state to begin with, right? But just focus on the transition. Now what I want to do is, again, anything that was off initially is now on, and anything that was on initially is now off. To do that, I need to inject an excitatory or inhibitory control signal into every single node in the network to allow this state transition to happen, because I can't take advantage of any of the network structure. I really have to just force it from the top. Okay, so what this helps us to think through, again, from an intuitive perspective, is that the ease or difficulty of a state transition in a networked system depends on the network, it depends on the structural connections, it depends on where activity can flow. So we can think through that kind of explicitly using dynamical models, but I sort of want to go from this simple model that I've shown you already to expanding out. How broadly can we apply this? How can we change the model to make it relevant to other problems, etc. So to do that, I'm going to ask, what are the inputs and the outputs of a network control model for a brain? Well, the inputs, I think I've already specified, are two. First is a mapping of the network, and second is a model of the dynamics. And then what are the outputs of the network control model? Well, typically the design of the perturbation. Where are the control signals? What is their time evolution? How spatially distributed are they? Into which region should they be injected, etc. So let's think through these three different pieces, the two inputs and the one output. So for the input, what is a map of the network? Well, I've been talking today mostly about the human data. So for us in studying humans, it would be a diffusion tractography, typically. However, if we're thinking about other animals, we could use other techniques. For example, trap tracing in macaques or electron microscopy for cellular level circuits. Now let's think about the second input, the model of the dynamics. The one that I showed you initially is something similar to this top one, which is linear and time invariant. What this is saying is that the change in the state of the brain is equal to the previous state times the adjacency matrix and then plus the control. So that's basically what I had shown you previously. However, this also states that it's possible that what we can measure, y of t, is different than the true state of the brain, and that there's some mapping between them. But we can increase the complexity of that model in a couple different ways. The simplest next step is to add in time variation for the adjacency matrix, so allowing the adjacency matrix to change with time, but also allowing the B matrix to change. And that's the one that is indicating where stimulation is being applied or where the control inputs are being injected into the system. We can also allow for a time varying transform from the state of the brain to what we can actually measure, assuming that what we're measuring is different than the true state. And then from there, we can continue down along this dimension of complexity, eventually hit fully nonlinear models. Those are more complex, but they're also can be really powerful. So there are fewer, if you really like math that you can do on paper, the nonlinear models are hard. We can do a lot more here and build intuitions. However, for some particular problems and where simple analytical tractability is not necessary, then the nonlinear models can be really great. Okay, so that has taken us through the mapping of the network and the model of the dynamics. And now I want to spend a couple minutes thinking through the outputs. I stated this pretty simply as the design of a perturbation, but I want to ask, what kind of perturbations, what kind of designs, what kind of metrics can we calculate from this approach? So we can summarize them pretty simply, from simpler to more complex. So here, for example, the simplest one is called the impulse response. So that's a system's output when presented with a brief input signal. So if we take the brain, we ping it once in one region, and we see how all regions change in their activity and summarize that, that's the impulse response. By contrast, a controlled response is not just pinging once. It is a system's response to some controlling input UT from some initial state. So an explicitly defined input that is seeking to push the system to in a particular direction. We can also assess the controllability of the system. So a system is controllable if there is a control input that brings the system from any initial state to any final state in finite time. And so you can estimate that for a variety of different systems, but also for brain systems. But I think that the output that I'm most excited about and that I think is really relevant to trying to understand and personalize and guide the development of stimulation interventions is this final one. So achieving desired state transitions through minimum energy control. So in particular, what I want to do is to design a control input UT to minimize the control energy and possibly other factors to drive a desired response. And when I say desired response, what I mean is moving the system from some existing activation state to a preferred activation state. And that this trajectory can require very complex control input and may have other constraints. For example, when we're using stimulation, we want to minimize the stimulation so that we won't heat the tissue, for example. There are additional constraints, sort of the length of time that you're going to stimulate and a variety of other things. So this is the one I'm really most excited about. Now, we can use that approach to quantify the ease and difficulty of state transitions. I've spoken to you so far about that there are easy and difficult state transitions, but I just showed you pictures to build an intuition, right? So if we wanted to quantify whether and how much a state transition is easy versus difficult, how might we do that? Well, we typically do it by defining a cost function that allows us to assess the minimum energy trajectory from an initial state to a desired final state, given the network underneath. And this is the cost function that we typically used. So this is a minimization of two factors. One is the distance of the current state X from the target state. And so what does that do? That basically allows us to follow this trajectory in as straight a manner as possible through whatever n-dimensional state space the system is moving. So we want it to be relatively straight so that the system is not pushed in kind of wildly different directions, perhaps hitting pathological states along the way that may be detrimental to the system's function. So we try to keep it as straight as possible. And then in addition to that, we minimize the energy that has to be injected. So use of kappa is this control input that I've been talking about. And we want to minimize that for two reasons. One, I just mentioned in the context of actually stimulating a brain, you want to make sure that you minimize certain features of it so as not to heat the tissue. But also when we apply the same approach to an understanding of intrinsic control properties like cognitive control, we want to minimize this energy because we know that the system is working under strong energetic constraints in terms of the biology. So I do also want to point out that there is a variable here, that rho, which controls the relative importance of minimizing energy versus minimizing distance. And you might ask the question, well, how would you choose rho? Do we know how important it is for a brain to minimize the energy of the transition versus to minimize the distance that is traversed by the system through the state space? The answer is no, we don't know, or we don't know now. So what we do typically is to have rho equal 1. So have these two components equivalently impact the simulation, but then we also demonstrate that the results hold over a variety of rho values. But if there are contexts, tasks, or that in which, or maybe organisms, or other ways in which we could determine which of these is more important quantitatively from data, then we could use that in the model. Okay, so that just brings me to the summary slide. What have we done over the last few minutes? We started with the statement that a brain is a networked biological system. And then I talked about the structural network constraints in the brain on how activity can flow through the system, thereby impacting its response to stimulation. At the beginning of the talk, I focused on explaining a couple recent efforts that have expanded the investigation of structural constraints. They included using increasingly sensitive measurements, developing data-informed computational models, and then embracing a conceptual shift in explanations for the efficacy of stimulation. Together, those expansions are providing us with a richer understanding of the structural constraints on both normative neural function and the response of the system to stimulation. I showed you one particular example of the importance of structural connectivity and explaining the response of the brain to stimulation. And then I walked through one particular example of a data-informed computational model. For anybody who's interested in using that data-informed computational model to study state transitions that you care about, in particular task scenarios, we do have a primer on the technique, a practical guide, a GitHub repository, and a review. The people whose work I focused on today, other than Val Sidner, who led the example TMS study, included work from Teresa Carr, from Jason Kim, from Jenny Stiso, and from Pragya Srivastava. So with that, thank you so much for listening. Time for questions.
So thank you, Professor Bassett, for that fantastic and very, very insightful talk. So I think we'll open it up to questions. Are there any questions from the audience?
So there is a lot of control systems terminology, right, in the formulation of the problems, the dynamics, but also even in the control function. You had typical quadratic state cost, and then quadratic cost for the control. So my question is that because we understand how to handle the systems? Is it mathematical expediency that leads to this type of models? Or there is evidence, experimental evidence that validates this type of models?
Yeah, that's a really great question. So I would say that the initial formulation was definitely pragmatic, that it allowed for us to do analytical work that was helpful in gaining some intuition. But we also have a study that we published in Cell Reports where we take this model and say, if we have the brain in a particular state and then stimulate it for real, like with actual stimulation, can we predict using the model where the state will go and to what degree? And so what we can show is that we can predict the next state better than chance. But there's, I think, still a lot of variance left unexplained. So to me, what that indicates is that the simple model has some practical validity with actual neuroimaging data and actual stimulation data. However, I do think that there are expansions to the model that make a lot of sense moving forward. One of them is this expansion to the time varying adjacency matrix. So I mentioned that that would be really important in the context of synaptic connectivity, where there are going to be changes in synaptic strength with time. But we also see this in the context of effective patterns of connectivity in human neuroimaging at the large scale. So the example that comes to mind right now is in the context of epilepsy and epileptic seizures. In the course of an epileptic seizure, there are typically 3 periods that the brain goes through. The first one is the propagation of the seizure, the next one, initiation of the seizure, then propagation, and then termination of the seizure. In those 3 different stages, there's a different effective pattern of connectivity between brain regions, and we can extract that using measures of effective connectivity. Those 3 different patterns of effective connectivity then impact where you would want to inject stimulation if you wanted to quiet the seizure during the initiation phase, during the propagation phase, or during the termination phase. So we have a paper that came out in PNAS last year, kind of illustrating how to do that modeling with the time varying adjacency matrix and how control would change for the seizure dynamics.
So that was great talk. And I love the models, actually, because I've been thinking about these. And so maybe I can connect the 2 parts of the talk. I'm very interested in the extent to which neural connections that make the brain work are resilient. And you've got your fiber bundle that's quite interesting, and the density of fibers. And one of the things that I'm interested in is how resilient are these connections to loss of function in some of these connections. And yeah, I've been thinking about that exactly in terms of linear systems and looking at different structures similar to the ones you have, where you can produce resilience if you have enough inputs and you can tolerate failure. So I'm just curious about, what you observe in the actual neuroscience. I mean, does disease and or injury affect loss of fiber density, and does that have behavioral consequences?
Yeah, definitely. There's the work that comes to mind right now is work from John Detry on periventricular white matter and specifically lesions in the periventricular spaces. And those are places where there's a density of structural connectivity, and lesions in those spaces impact cognitive function quite broadly because of the density of structural connections, and not density in one direction, but in kind of many directions, complex patterns of connections or directions. So that work suggests to me that resilience is going to depend a lot, I think, on the complexity of the area where the lesion occurs. And how many different other regions the connections go to, the fibers go to from there.
Dani, we have a question from the online audience. You briefly mentioned cognitive control. Could you expand on how you think about using network control approaches to understand some cases of cognitive control?
Yeah, absolutely. Thank you for asking. So we are really interested in using these ideas to study cognitive control in humans because there's a set of brain regions that are called cognitive control areas in the executive system, which are thought to exert control on other brain regions to enable the guidance of behavior, including, for example, inhibiting inappropriate behavior, but also supporting complex decision making. So we wondered how those brain regions do it. How is it that they play a role in control? And one possible explanation for that function of those regions is that they are connected to other brain regions in a way that allows them to use signals effectively to alter activation of other places in the cortex. And so that's something that we have studied in a couple different ways. Maybe one paper that I will mention if you're curious in this space is a paper from Jiaju Q, and Ted Satterthwaite is a senior author. It was published in eLife, and it focuses on understanding how the frontal parietal executive system can be activated as a human moves through periods of development. So the individuals who were being studied ranged in age from 8 years to 22 years. And what we asked is, how would each of those brains, which differ in their pattern of structural connections, move from no activation to activation of frontal parietal cortex, which is involved in cognitive control and executive function. And what we found is that the energy required to make that transition, to activate frontal parietal circuitry, changes with age. So it depends very heavily on how the white matter is changing over that developmental time period. And in particular, what we saw is that older youth in this cohort were able to activate frontal parietal circuitry with less energy, so with less control input than the 8-year-olds. And that was purely explained by changes in the white matter, the pattern of white matter connectivity. So what that suggests to us is that cognitive control is supported by the pattern of structural connections that exist in the brain, and that pattern of structural connections changes over development in a way that allows cognitive control to happen more easily. We also found that for those people who could activate frontal parietal circuitry with less control energy, they're also the individuals who scored more highly on a battery of tests evaluating executive function. So that suggests to us that our control energy measure is cognitively relevant, directly correlates with individual differences in executive function.
Thank you. Are there any other questions from the audience?
Hi, that was really great. You mentioned the advances that have happened in the precision of diffusion imaging in particular. And of course, we can expect more such advances and methods get more precise. I'd be really interested in hearing your perspective on kind of as the methods stand now, how does the kind of current level of precision or imprecision, what we can and can't measure, kind of play into some of the higher level network characteristics that we're interested in?
Yeah, that's such a great question. I think that I'm going to answer that by telling you what I want for Christmas, which is that I would love for these techniques to be able to show me more about the directionality of connectivity. And that's what I want for Christmas. But basically, that a lot of these techniques are, because most structural connections at the very large scale are bidirectional, largely, these estimates are going to be pretty good. But I think it's going to matter a lot where they're not, where the directionality matters. And so that's where I think the major limitation lies, for me anyway. And from what I see from the controls literature in other systems, the directionality can really matter a lot. And so yeah, that's where I think we need more.
Thank you for the great talk. I have a question about how do you take into account of the perturbation-induced change in the transition matrix A in terms of you're estimating the transition based on the original natural connectivity, and particularly if the perturbation itself would induce some imbalance in excitability of some connection, which could be a form of representation for some type of information.
That's interesting. If the perturbation drives a change in excitability, we would want to incorporate that not necessarily in A, but in B. So the B matrix is one that is accounting for, that sort of modulates how that region responds to the stimulation and then allows that stimulation to propagate elsewhere, right? So the B matrix is where that's going to be. So if we know something about how TMS is, or any stimulation is altering excitability, you would put that in the B matrix. And from there, I want to riff a teeny bit, because there's also some really beautiful work that is now showing that variations in the density of neurotransmitters in different areas can and does change, obviously, the response of areas to different exogenous inputs, and that you can also model that really beautifully by putting in the B matrix. So the B matrix along the diagonal can include information about the density of particular neurotransmitters that are relevant for that task in kind of explaining how that region responds to exogenous input from another area or from external stimulation.
Thank you.
Are there any other questions?
Thank you for the talk. I have a question, but I'm not sure if it's relevant. So is there any sort of correspondence or connection between these transition, like modeling and like systems of Hamiltonians people work on from physics?
And what kind of what?
Hamiltonian systems, like from physics?
I am not pulling up a direct map right now, but feel free to expand.
It's just like a similar property modeling technique that people work on, like in physics, like, and maybe like in modeling like quantum systems and things like that. But I'm not sure like whether it's related or connected. So just a wild question.
Yeah, I think where my mind is going right now is just there is a lot of work using, for example, icing models,
right.
and understanding control signals being applied to icing systems.
Icing.
To, for example, maximize influence, or to impact the sparsely connected regions versus the heavily connected regions. So there is a whole set of literature there. The question is kind of how to map from those kinds of models, like icing models, to neural systems. I think more could be done across those lines.
Did you have any references in this link, or?
No, but I can either send it to you or give it to you right after this.
Thank you.
Yeah.
Thank you for the talk. I'm curious about when you're making the analogy between a graph system and the physiology in your Laplacian or your adjacency matrix, how do you determine a node versus an adjacency? Is it like, for example, a series of fibers? Because I assume a neuron eventually is going to be the state of the art, but is it like a threshold bundle of fibers, or is it just like an arbitrary thing right now, or what exactly is it?
Yeah, that's a really good question. There are a variety of ways that the neuroscience community is defining brain regions right now. And I think it's an open question, which of them is most relevant for which kinds of questions. But just to give you some examples, some approaches consider just the cytoarchitecture, so the kinds of neurons that are present, the density of those kinds of neurons, and the kinds of interneuronal connections at the very small scale. So that's one way of doing it. And cytoarchitecture varies significantly over different parts of the brain. And so you can use that to set boundaries between regions. There are also efforts to use functional information, like for example, a resting state fMRI scan can be used to isolate sets of voxels that are responding really similarly over a long period of time. So that's a resting state based version. Then there are also ones that incorporate some information about structural connectivity. And there are some that combine these different kinds of information and try to say what's consistent across them, what's different across them. So the partialations vary significantly. What we try to do in our studies is to demonstrate, typically in the supplement of the paper, that our findings are robust to variation in the parcellation. What we often find is that it's not always robust across different spatial resolutions of a parcellation. So if you cut the brain into 50 pieces, or you cut the brain into 5,000 pieces, you sometimes don't get the same answer. And I think that that makes sense because the specificity of and spatial resolution is actually critical for understanding how the system is organized. So yes, but you can still construct several different parcellations at the same spatial resolution and demonstrate that your results are consistent.
Yeah, that would be very interesting because the atlas does keep changing, right, over time.
There's a proliferation of atlases.
Yeah,
yes.
Yeah.
Yes. Thank you. Any other questions? Are there any online? Yeah. So with that, so thank you for the audience for a great discussion, and let's thank the speaker one more time. So before we all adjourn, we welcome you to attend the next Hariri Institute sponsored MLX Med event, which is coming up on May 17th. It's a virtual event. So Emre Kitcheman from Microsoft is giving a talk on extending a causal analysis suite for health analyses, capturing and validating critical assumptions. So for more information and the link to registration, please visit the Hariri Institute's Eventbrite page. And otherwise, have a great weekend.